This conference will now be recorded.
Good evening, I'm Dr. Divya from TeenEinstein. So now we are in the 6th chapter, molecular basis of inheritance. So in this chapter, we are mainly discussing about DNA. What is DNA? Deoxyribonucleic acid. And then another one which we are discussing about RNA, ribonucleic acid. So these are the two types of nucleic acid found in living system or living organisms. So in most of the organisms, DNA is acting as the genetic material. So that everyone will be knowing that DNA is the main genetic material, or mainly DNA is seen as the genetic material. And RNA is also acting as a genetic material in what and all, in some kind of viruses you can see RNA as a genetic material. And RNA that works mainly as a messenger. What is the main role of RNA? It is mainly working as a messenger. So then we discussed about the structure of polynucleotide chain, and which are the three components of that nucleotide. Three main components of a nucleotide. Nitrogenous base. Which are the three main components of a nucleotide? Nitrogenous base, a pentose sugar, a phosphate group. These are the three main components of a nucleotide. Nitrogenous base, pentose sugar, and a phosphate group. Then we learned about purines and pyrimidines. What are they? Purines. Which are purines? Adenine and guanine. Then pyrimidines. They are cytosine, uracil, and thymine. Uracil you can see in RNA. Just I am giving. I'm explaining everything shortly because we need to know about all these things. Then only we can understand the explanation of today's class. So nitrogenous bases, pentose sugar, and phosphate group. That is the three main component of a nucleotide. And purines and pyrimidines we have learned. Purines means adenine, guanine. Then pyrimidines, which are the pyrimidines? Cytosine, uracil, and thymine. In that uracil we can see in RNA. Then we discussed about the double helical structure of DNA, right? So in that double helical structure, it is made up of two polynucleotide chains. In double helical structure, we can see two polynucleotide chains, and these both chains, they are having anti-parallel polarity. That means one from 5' to 3' end and another from 3' to 5' end. So double helical structure, it is made up of two polynucleotide chains, and both chains, they have anti-parallel polarity. What do you mean by anti-parallel polarity? One from 5' to 3' end and another chain from 3' to 5'. So these strands are paired through hydrogen bonds. With which bonding? With hydrogen bonds they are paired. And both polynucleotide chains, they are coiled in a right-handed fashion. You can see here in the DNA, they are coiled in a right-handed fashion. Right turn. Okay. In a right-handed fashion, they are coiled. And the plane of one base pair that stacks over the other one. In the double helix, how you can see? The plane of one base pair, the plane of one base pair stacks over the other one. And that forms the double helical structure. Okay. So, in double helical structure, it is made up of two polynucleotide chains, and both the chains are anti-parallel. They have anti-parallel polarity. That means, what do you mean by anti-parallel polarity? One chain is from 5' to 3' and another one is from 3' to 5'. Exactly opposite way. So these strands, they are paired to hydrogen bond, and Both polynucleotide chains, they are coiled in a right-handed fashion, and the plane of one base pair stacks over the other one. So that's how you can write for your board exam, you can write this as the double helical model of DNA, the structure of double helix. Then Francis and Crick, they proposed the central dogma in molecular biology. Important question for NEET exam. Who proposed central dogma? What do you mean by central dogma? DNA, RNA and protein. So who proposed this? Francis and Crick. They proposed the central dogma in molecular biology. What is central dogma? DNA to RNA and then to protein. From DNA, RNA is forming and RNA is forming the protein. Okay, protein is the final expressive form. Then we learned about Griffith's experiment, and he discussed the transferring of genetic material through the same experiment. How genetic material is getting transferred, that's what he tried to explain through his experiment, Griffith. So transferring of genetic material, that was explained by Griffith, but he didn't explain the biochemical nature. Biochemical nature of the genetic material was not explained by Griffith. And who explained about that biochemical nature or the structure or the nature of the DNA? Biochemical nature of the DNA. Avery, MacLeod and McCarthy. Note down all these scientists' name because you will get confused. In this chapter, you are having so many scientists' name to learn, and they have explained different things. So note down Avery, MacLeod and McCarthy. They determine the biochemical nature of this transforming principle. In Griffith's experiment, he explained about that transferring of the genetic material. Transferring of this genetic material, it is explained by Griffith, but the biochemical nature of that transferring material, which material is getting transferred, the nature or the biochemical nature of that material is explained by Avery, MacLeod and McCarthy. Then another experiment is Hershey and Chase. They conducted another experiment, that is to prove DNA is the genetic material. Who proved DNA is the genetic material? Through which experiment? Hershey and Chase. Through their experiment, they proved DNA is the genetic material. So what they did in their experiment? They started their experiment with the culturing of bacteriophages. Bacteriophages are nothing but virus that infects bacteria. So what is bacteriophage? Virus that infects bacteria. So one set of bacteriophage or virus, it was cultured in a medium of radioactive phosphorus. That was the first step. In a medium of radioactive phosphorus, one set of bacteriophage was cultured. Then another set was cultured in a medium of radioactive sulfur. One was in radioactive phosphorus. Another set of bacteriophage was cultured in a medium of radioactive sulfur. And they observed. What was the observation made by them? So they observed that in the first set of bacteriophage, they have radioactive DNA. In the first set, they have radioactive DNA. But there was no radioactive protein. Why? Why is it so? Because DNA, in DNA, which one is there? Phosphorus. DNA is the phosphorus based compound, but protein don't have phosphorus, right? First set was with which one? First set was with radioactive phosphorus and another set was with radioactive sulfur. So in the first set, they have radioactive DNA. Why they have the radioactive DNA? Because phosphorus is a part of DNA, or DNA is a phosphorus based compound. But protein doesn't have DNA. Protein doesn't have phosphorus. So radioactive phosphorus came in which one? Radioactive phosphorus came in DNA because phosphorus is a part of DNA. It is not a part of protein. So in DNA, they can see radioactive phosphorus in that experiment, or they saw the presence of radioactive phosphorus in that one. But in the second set was with radioactive sulfur, right? So in the second set, the bacteriophage or the virus that was not having radioactive DNA, instead of that was having radioactive protein, because sulfur, that is a part of protein. But no radioactive DNA was found in the second set. So E. coli bacteria was the host for that infection. You have to learn about this experiment very clearly. You should not get confused, because you may get for your board exam and you may get many neat questions from this part. Okay. So E. coli bacteria was the host for the infection. So they did many number of blending, centrifugation, then different, and they allowed these viruses for the infection. They allowed to infect the bacteria. And what was the result of that? E. coli bacteria, they were infected by radioactive DNA virus. Okay, but the one where in the second set we said it was protein, right? But one that infected by radioactive protein virus, they were not radioactive. Here with DNA, that was radioactive. Here with protein, it was non-radioactive. So how they concluded their experiment, or how they concluded their result? The virus that had radioactive DNA transferred their DNA to the bacteria. But the virus that had radioactive protein did not get transferred to that bacteria. So what do you mean by that? We can say that DNA is the genetic material. DNA is getting transferred from one to another one, but not the protein. So up to this we have discussed in last class. So in today's class we are going to discuss about properties of this genetic material. So first we are going to discuss about properties of genetic material. So before learning this, you should be thorough with, or you should be clear with the structure of DNA, then this Hershey's and Chase experiment, all these things you should be clear. Okay, so now we are going to discuss about properties of this genetic material. Genetic material, DNA, and in some organism you can see, most of the eukaryotes you can see DNA as the genetic material, but in some kind of viruses and all you can see RNA as the genetic material. So from Hershey's and Chase experiment, it's very clear. It's very clear that DNA is acting as the genetic material, right? But you can see in some kind of organism, like some kind of viruses and all, RNA is acting as a genetic material. For example, which one we can say? Tobacco mosaic virus. In that, note down this. In tobacco mosaic virus, RNA is acting as a genetic material. So we can see that DNA is the main genetic material, or the predominant genetic material in most of the organism. But in few organisms, you can see RNA as the genetic material. So RNA, mainly how RNA works? RNA mainly it is working as a messenger. The main role of RNA is it is working as a messenger. Okay. So DNA as the genetic material was unequivocally resolved from Hershey and Chase experiment. From Hershey's and Chase experiment, it's very clear that DNA is working as a genetic material. And it became an established fact that it is the DNA that acts as a genetic material. So it's very clear that DNA is acting as the genetic material. And in some kind of viruses like tobacco mosaic viruses and all, RNA is the genetic material, or RNA is acting as a genetic material, for example, in few cases. For example, tobacco, mosaic virus. So in most of the organism, you can see DNA as the predominant genetic material, and RNA performs dynamic function of messenger. So mainly RNA is acting as a messenger, and there is some kind of changes or differences between the chemical structure of these two nucleic acid molecules. In the structure of DNA and RNA, you can see some kind of differences also. So DNA is acting as the main genetic material in most of the organism. Only in few cases you can see RNA as the genetic material. In the case of some kind of viruses like tobacco, mosaic virus and all, you can see RNA as the genetic material. Then here is the properties of genetic material. So to qualify as a genetic material, there are some important properties. There should be some important properties for that particular material, or that should fulfill some kind of characters or some kind of properties to act as or to function as a genetic material. So let's see each and every point. So the first point is a molecule that can act as a genetic material that should fulfill some kind of qualities, or they should have some kind of some criteria. What are they? First point is it should be able to generate its replica. That should be able to generate its replica. That should be able to form the copy or replica. So the genetic material that should be able to generate its own kind. That means the process of replication. That is the main thing. The process of replication here they are meaning. So both the nucleic acids, both DNA and RNA, they have the ability or they have the capacity to do the duplication or replication, to form the copy. But in some other molecule like protein, they don't have this function or they don't have this character. That molecule in living system like protein, they are not fulfilling this criteria. Okay, only this DNA and RNA, the genetic material, that can act as a genetic material because that is fulfilling some characters. What are they? First point is it should be able to generate its replica or the copy. So the genetic material should be able to generate its own kind. That means the replication process, both the nucleic acid, that means both DNA and RNA, both are nucleic acid, DNA and RNA, they have the ability or they have the capacity to do the duplication. Okay, but in some molecule like protein or some other molecule in living organism, you can see protein and some other fatty acid, protein, all these things. But all these, they are not fulfilling this replication or they are not fulfilling this duplication of that. Okay, so DNA that is suitable or that is fulfilling the first rule. That means that should be able to generate its replica or replication. And the second point is it should be stable chemically and structurally. Okay. So the second point is it should be stable. What are all the different criteria that should fulfill or the genetic material should fulfill? The second point is it should be stable chemically and structurally. What do you mean by this? The chemical stability or structural stability. That means genetic material should have a stable nature. So that should not change with the different stages of life cycle. In an organism will pass through different stages of life cycle, right? So during these stages, the genetic material should not change or the genetic material should not change the features. Okay, that should have a stable feature. So that should not change with different stages of life cycle and that should not change with the age. While changing the age, that genetic material should not change the characters or during the change of physiological changes of an organism also, that should not get changed. So in Griffith's experiment, stability of this genetic material, it was very clearly explained, or it was proven in Griffith's experiment. Okay, so according to the criteria which must be fulfilled by a genetic material, first one is that should be able to generate its replica, or it should be able to generate or form its copy, and that should be stable chemically and structurally. Chemically and structurally, this should be stable. It should not change with age or physiological changes, or that should not change during different stages of life cycle also. Okay. Then the third point is it should be, or it should provide the scope for slow changes that are required for evolution. Right? What do you mean by that? That should provide a scope for slow changes or mutation that are required for evolution. What is the meaning of that? That means that genetic material should show the mutation or slow change, that should change very slowly. There should not be a sudden change. Sudden changes are happening means when it is getting replicated or when a copy is forming, that will be entirely different, right? So there should not be a sudden change. There should be a slow change which shows the mutation or variation from the previous generations and all. So that makes the evolution, but that evolution should be in a slower ratio. It should not be a sudden change. It should happen very slowly. So the genetic material should show changes of slow rate changes, that should show, and that should be able, that means that should undergo mutations. That means both DNA and RNA, they are able to mutate. Okay, this RNA, you know, RNA is unstable, and you know that in RNA we can expect these changes in a faster rate, right? Because RNA is unstable, so that mutation also will be in a faster rate. So viruses having this RNA or RNA as a genetic material, and they are having very short life cycle that can mutate in a faster way. That's why now we are saying mutations in COVID. In COVID, RNA, in coronavirus, which one is the genetic material? RNA is the genetic material, right? And RNA is usually that is unstable and that is undergoing sudden changes. So mutation rate is more. So now we are telling different types of COVID viruses are there, or mutated forms are there. That's why RNA is unstable and mutation rate is high compared to DNA. That's why now different mutated forms of COVID are present. So hope you noticed that all these changes in newspaper daily, you will get so many mutated forms are there, different mutated forms have discovered. So hope now you understood what is the reason of that mutation. So they have evolved in a faster rate. That evolution or that changes that will happen in a faster rate compared to DNA. If the genetic material is RNA, it is unstable and there is a chance of mutation, sudden mutation in that. Okay, the mutation rate is more. Then it should be, next point is it should be able to express itself in the form of Mendelian character. Okay, that should be able to express itself in the form of that Mendelian characters. How that is getting expressed, that should be able to express itself as the Mendelian characters. Then, so these are the properties of a genetic material. So with what are all the properties? Molecule that can act as a genetic material that must fulfill the following characters. Character 1 is it should be able to generate its replica, or that is replication, another copy. It should be able to form the copy of that. Then it should be stable chemically and structurally. That should have, the genetic material should have chemical stability and structural stability. If the chemical stability or chemically it's not stable and structurally it's not stable, means it's impossible to make a copy. When it is making a copy, it will definitely that will have some kind of changes if it is not stable. If it is unstable chemically and structurally, it's unstable means that copy or that replicated one that will be having some kind of difference from this, right? And third point, it should provide the scope for slow changes that are required for evolution. There should be a scope of slow changes, not in a faster rate. They should have a slow rate of changes. And finally, after a few years, that may lead to evolution or some different kind of organism that may form. Some kind of changes may happen and it will evolve. But if that is in a faster way or sudden changes are happening, means it is very difficult to manage. And it should be able to express itself in the form of Mendelian characters. Got it? So these are the characters one genetic material should have. Next. So now we know that both DNA and RNA, they have the capacity to duplicate themselves. But you know that proteins don't have, or they don't have the capacity to replicate, or they don't have their own, or they are not fulfilling all these characters of the genetic material. So we have already told that genetic material should be stable and that should not change with age or physiological change or the changes in the life cycle of an organism, right? So if you are heating that complementary strands, that will get separated. In a DNA, if you are providing more heat, that complementary strands or the strands which are attached, both strands will get separated. Then, if you are providing the suitable condition like earlier or the normal condition, again these strands will come together or they will get attached together. Okay. So, just check point by point. If one examines each requirement one by one, both the nucleic acid, DNA and RNA, they have the ability to direct their duplications. Okay. And the proteins fail to fulfill first criteria itself. Proteins, they won't fulfill the first criteria. And the genetic material should be stable enough not to change with different stages of life cycle. We have already told that genetic material, the first criteria, that should not change always. Okay, they should have a proper chemical structure, then that should not get changed always. So during the changes of life cycle, that genetic material should not get changed. That should not get changed with life cycle, age, or with the change in physiology of organism. Okay. And stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself. So in Griffith's transforming principle, the main thing was proven. What was that? The stability of the genetic material. It was proven during Griffith's transforming principle. In that experiment, they tried to prove the stability of the genetic material. Then, two strands being complementary, if separated by heating, come together when appropriate conditions are provided. Two complementary strands of a DNA, they'll get separated if they are heating. If they are getting heated, that two strands, complementary strands, will get separate. They'll get separated, and when the suitable conditions or the proper conditions are providing, again they will come together and they'll get attached. Okay, here these bonds will be broken when we are increasing the temperature or heat. Okay, while during this PCR reaction and all, we are applying this technology. We'll give high temperature to break these bonds. Then these strands, both strands will get separated, and these strands again it will get, with the help of the reagents which we have added, or we are adding to that chemical reaction, with the help of those reagents, that will get replicated. So many copies of this DNA, they are creating so many copies of DNA with the help of the reagents which are added to that reaction. That's how we are conducting this PCR. Okay, polymerase chain reaction. Now, with the help of this PCR only, we are detecting the presence of COVID. Okay. So, DNA and RNA, they have the ability to direct their duplication. The protein fails to fulfill this duplication or the replication. They don't have the capacity to replicate. Then the genetic material should be stable enough and they should not change with the different stages of life cycle like age or with the physiological changes of the organism or during different life cycle of organism. So that should be stable. And stability that was proven with which experiment? Griffith's transforming principle. In that experiment, they proved the stability of genetic material and the two complementary strands that will get separated by heating, or if you're providing heat, that those strands will get separated, and these strands, when we are providing the normal condition, they will come together and they'll get attached together. Okay. Next is in the second position. Here in the second position, the presence of OH group in every nucleotide of RNA. In RNA, in second position, presence of OH group. In the nucleotide. Okay, that makes RNA unstable. The presence of that OH group in the second position, where in the second position that OH group is present in the nucleotide of RNA. So that makes RNA unstable, or that makes RNA reactive. So the RNA will become reactive, and that makes this RNA unstable, or that RNA becomes degradable. But DNA don't have this OH in the second position. So it is less reactive and DNA is more stable. So when compared to RNA, DNA is more stable. Why? In the second position of each nucleotide, RNA is having OH group. That makes RNA more reactive and RNA is becoming unstable and it is easily degradable. So DNA don't have this OH group, so that becomes stable, or it is less reactive and more stable. So when compared to RNA, DNA is more stable. So in both DNA and RNA, DNA works as a better genetic material. Why? DNA is having more stability and it is less reactive. Okay, so that is more stable. So changes are very less compared to RNA. So in both DNA and RNA, DNA is acting as a better genetic material due to the stability and it is less reactive. Okay. So the presence of OH group at every nucleotide in RNA, that is the reactive group, and makes RNA liable and easily degradable. That OH, presence of that OH group makes RNA liable and easily degradable. Then RNA is catalytic, so it is reactive. Therefore, DNA chemically is less reactive. DNA is chemically very less, or it is less reactive and stable when compared to RNA. Therefore, among the two nucleic acid DNA and among DNA and RNA, DNA is the better genetic material because of stability and less reactive. So, in fact, then one more point is the presence of thymine at the place of uracil, that also confers additional stability to DNA. I said in RNA you can see uracil. Instead of thymine, you can see uracil in RNA. But in DNA, thymine is present, and thymine that makes or that gives additional stability to DNA. So uracil, in the place of thymine, uracil will be present in the case of RNA. So that makes unstable. When it comes to DNA, in the place of uracil, thymine is present and that gives additional stability to DNA. Okay. Next, both DNA and RNA, they may undergo mutation. So mutation is faster in RNA because RNA is unstable and it will undergo sudden changes and it is getting mutated in a faster way or in a higher way compared to DNA. The rate of mutation is higher in RNA when compared to DNA. Okay, so those viruses which are having RNA as genetic material and they have very short lifespan, in them the mutation is faster. So the evolution or changes also in a faster way. That also will be in a faster way. So the virus is having RNA as the genetic material and they have a short lifespan. In the mutation is faster. So the evolution or the faster way. That's why in COVID, you can see, in the case of corona, you can see mutated forms, more mutated forms. Okay. So you know that DNA codes for RNA, then RNA codes for protein. First DNA, from DNA, RNA is forming, then RNA is forming protein. Right. So here, RNA can directly code for, or they can directly give protein. Right. In the case of DNA, first DNA should form RNA. Then this RNA only can give protein, right? RNA only can form protein. But in the case of RNA, there is no intermediate step. From RNA, you will get directly protein expression, right? So here RNA can directly code for or they can directly synthesize protein. So the expression of character, it is easy for RNA, right? The expression of character, it is very easy for them, which are having RNA as the genetic material. But in the case of DNA, they are depending on RNA, right? DNA should depend on RNA because first DNA should code for RNA and then only that RNA will code for or that RNA will synthesize protein and then the protein will get expressed, right? So the DNA is more stable than RNA. So the DNA is preferred for what? Preferred for the storage of genetic information. So DNA is more stable than RNA. So DNA is more preferred for the storage of genetic information. But for genetic transmission, note down the point, for genetic transmission, which one is preferred? Genetic transmission, RNA is preferred. Okay. So both DNA and RNA, they are able to mutate. In fact, RNA being unstable mutate at a faster rate. RNA, it is unstable compared to DNA. So that will get mutated in a faster way. And the virus is having RNA genome and having shorter lifespan. They mutate and evolve faster. The viruses which are having RNA as the genetic material and which are having a shorter lifespan, they will get mutated because lifespan is short. So the mutation that will in the next generation, it is very soon, it will get expressed in the next generation or the changes will get expressed in the next generation in a faster way, right? And RNA can directly code for the synthesis of protein. RNA, if the genetic material is RNA, that next step is protein synthesis. There is no steps in between. So direct expression and the time taken for the expression of that character, that will be very easy, or that can the expression will be very easy compared to DNA. But DNA is dependent on RNA for the synthesis of protein. If DNA is the genetic material, first that should form RNA, then RNA only can form protein, right? So the DNA is dependent on RNA for the synthesis of protein. So the protein synthesis or protein synthesizing machinery has evolved around DNA, and both DNA and RNA, they can function as genetic material, but DNA is more stable, so that is preferred for the storage of genetic information. because DNA is more stable. That's why it is preferred for the storage of genetic information and for the transmission of genetic information. For the transmission, genetic information transmission, which one is better? Better. Next is about RNA. So RNA world, in that, which is the first genetic material? Do you have any idea? Which one is the first genetic material? It is DNA or RNA? Which is the first genetic material? RNA. RNA was the first genetic material. Why we are saying like that? Because processes like metabolism, then splicing, translation, or protein synthesis, all these are related to RNA. You know that protein synthesis is related to RNA. So the first genetic material, that must be RNA. So RNA works as a genetic material, and in some other cases it is acting as a catalyst also. So in some important biochemical reactions, which are the, in some cases, those reactions are catalyzed or catalyzed. What a catalyst will do? The rate of reaction and the energy, that energy of reaction, that will be less, right? So that more substrate and enzyme reaction will be more, right? So in such cases, processes like metabolism, then protein synthesis, all this are related to RNA. So in that case, RNA, in some cases, it is working as a catalyst also. Some important biochemical reactions are catalyzed by RNA, and that is not catalyzed by enzyme. The rate of reaction, it will get increased, or that is not by enzyme. That role is done by RNA. So in normal cases, enzyme is performing that role, right? In normal case, enzyme is doing that catalysis. But in some special cases, RNA is performing that role. Not only it is working as a genetic material, sometimes it is catalyzing the reaction also. So here in some cases, RNA is acting as a catalyst. Okay. So a catalyst that is having a reactive nature, that is participating in that reaction. So that is having a reactive nature. So what happens in that case? It becomes unstable, or though that RNA which is acting as a catalyst, that is having a reactive nature, so that definitely it will become unstable, right? So then DNA, it is evolved from RNA, and some chemical modifications, that modifications made this DNA more stable, right? So DNA is formed, or DNA is evolved from RNA, and DNA is modified form of RNA. What we can say, DNA is the modified form of RNA. These modifications, the modifications in RNA, that makes that DNA more stable, or some kind of modifications happened in that RNA that makes that DNA more stable. Okay. So RNA was the first genetic material, and essential life process like metabolism, splicing, translation, etc. These are evolved around RNA. Then RNA used to act as a genetic material as well as a catalyst. Okay. For some biochemical reactions, that RNA is acting as a catalyst, and RNA being a catalyst was reactive and hence unstable. Why RNA is becoming unstable? In some cases, it is acting as a catalyst. So catalyst means it will participate in that reaction, so it becomes reactive. So that reactive forms, it will become unstable. Definitely, it will become unstable. So DNA that has evolved from RNA with chemical modifications. So DNA has evolved from RNA, and during that evolution or that evolving process, that that has undergone some kind of chemical modifications, and that modifications makes this DNA stable. Okay. Then next is DNA replication. DNA replication. So who proposed that DNA replication? Watson and Crick. So according to them, or their original statement, here you can see here the original statements I have given. So Watson and Crick, they proposed the DNA replication. So according to them, or according to their original statement, the first statement here you can see the original statements here. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Okay, that is the first statement. And the second one is the scheme suggested that the two strands would separate and act as a template for the synthesis of a new complementary strand. The two strands, here you can see the two strands of DNA, right? These two strands that can get separated, these two strands can get separated, and each strand can act as a template for the synthesis of new complementary strand. After separation, each strand will act as a template strand, okay, or a mother strand which can help for the synthesis of a complementary strand. Here you can see this complementary strand is base pairing, that is complementary. Okay, adenine will pair with thymine and guanine with cytosine. So that complementary base pairing, based on that, they can form another strand. So after completion of replication, each DNA molecule would have one parental strand and one newly synthesized strand. So after DNA replication, how it will be? Each DNA molecule that will be having one parental strand and one newly synthesized strand. And this scheme was termed as the semi-conservative DNA replication. So what is semi-conservative? From that term itself, it's clear. Semi-conservative. Semi, half is conservative. Half of that is conserved, or that is the old one, and another half is newly synthesized. So semi-conservative DNA replication. In this, two strands would separate and that will act as a template for the synthesis of a new complementary strand. And after the completion of that replication, each DNA molecule will have one parental strand and one newly synthesized strand. And this scheme was termed as semi-conservative DNA replication. Okay. And next, DNA, it is capable of self-duplication. And they said that Watson and Crick in their statements, another one, next one is DNA is capable of self-duplication. This DNA, deoxyribonucleic acid, they can duplicate themselves, or that is capable of self-duplication. Then all living beings have the capacity to reproduce because of this characteristic of DNA. Okay, so all living organisms, because of this duplication or this duplicating character of the DNA, all living beings have the capacity to reproduce. If they are reproducing, their copy will be formed, right? There won't be so much changes, because half of the character, it is getting separated. One DNA strand is getting, both the strands are getting separated, and half strand will be there, another half is newly synthesized. So the offspring or the children will be having the character of mother and father. That's why it is having the capacity to reproduce because of this characteristic of DNA. Then DNA replication is taking place in S phase of the cell cycle. In 11th standard, you have learned about cell cycle and cell division. In that, they have mentioned about S phase. Which is the S phase? S is the synthetic phase. Synthetic phase. In the synthetic phase, what is getting synthesized? DNA or the genetic material, that is getting synthesized in the synthetic phase, right? G1 phase, S phase, and G2 phase. These are the phases of a cell cycle, right? G1 phase, S phase and G2 phase. In that S phase, in the S phase, what is there? In that S phase, synthesis of DNA, synthesis or synthetic phase or S phase. In that synthesis of DNA is taking place. So at the time of cell division, it divides in equal parts in the daughter cells. So DNA is getting synthesized in S phase, and the time when cell is getting divided, that time that DNA is getting divided equally, or it divides in equal parts in the daughter cells. Got it? Then next is semi-conservative model of DNA replication. We have already told half of that will be there, another half is getting synthesized, right? So semi-conservative model of DNA replication. Without experimental proof, we can't accept anything, right? So there should be an experimental base for each and everything. So semi-conservative model of DNA replication. This semi-conservative mode of DNA replication was first proposed by Watson and Crick, right? We know that Watson and Crick only proposed that, but it was experimentally proven by Michelson and Stahl, who proved the semi-conservative mode of DNA with experimental proof. Who gave the experimental proof for this semi-conservative mode of DNA replication? Michelson and Stahl experiment, which experiment is that? Michelson and Stahl experiment. So what they did, and just before that I'll tell one more point. Michelson and Stahl, they gave the experimental proof, and in 1958 they did this experiment, or they gave this proof, and it was in E. coli. These points, please note down these points, because this is important for your NEET exam. This is in E. coli. In which one they conducted this experiment? In E. coli. Then another scientist, Tyler, and he did this experiment in Vincia faba. That is the experimental material, and that also in 1958. Okay, for that experiment, Taylor used radio tracer. Okay, radio tracer technique. That technique was radio tracer technique. Means the radio isotopes. He used radio isotopes, but Michelson and Stahl experiment, they used heavy isotopes of nitrogen. With the help of heavy isotopes of nitrogen, they proved this. Okay. So first, what they did, Michelson and Stahl experiment, they grew E. coli in a medium containing ammonium chloride. So, but in this ammonium chloride, the nitrogen is the heavy isotope of nitrogen. Here, the ammonium chloride in that N, nitrogen is heavy isotope of nitrogen. Okay, so they grow E. coli in a medium containing ammonium chloride as the only nitrogen source for many generations. This ammonium chloride was the only nitrogen source for many generations, but in that ammonium chloride, that nitrogen was with heavy isotope of nitrogen. That nitrogen was heavy isotope of nitrogen, and the result was that this N15 that was incorporated into newly synthesized DNA, and not only in DNA, the other nitrogen containing compounds also, it was having this heavy isotope of nitrogen, which and all compounds are with nitrogen. In all, this was the source. This heavy isotope of nitrogen, this was the one and only nitrogen source. So in result, all the nitrogen compounds, it was having this heavy nitrogen isotope. Okay, and this heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. Okay, so how we can differentiate or how we can distinguish this normal DNA from this heavy isotope DNA? By centrifugation and cesium chloride density gradient. Cesium chloride density gradient centrifugation. Through that we can make out the heavy isotope DNA and the normal DNA. Got it? Then they transfer the cells into a medium with normal ammonium chloride. This is with normal ammonium chloride N14. That is not with isotope N14 only. And took same sample. They took the sample at various definite time interval as the cells multiply. In different stages of multiplication, they took the samples. Okay, and they extracted the DNA from that. They extracted double-stranded DNA from that, right? So what they did? First, they provided that that isotope nitrogen, that N15 nitrogen source for many generation. N15, that is the heavy isotope of nitrogen. They provided this as a source of nitrogen. Then they transferred those cells into medium with normal nitrogen or normal N40. Okay, ammonium chloride. In that nitrogen N14 was present, and they took or they collected samples in various stages or various time intervals of that cell multiplication, and they extracted the DNA. They extracted the double-stranded DNA from that. Then what they did? In that cesium chloride density gradient centrifugation, they separated these samples and they checked the densities of that DNA. They checked the densities. So the DNA that was extracted from the culture 1 generation after transfer from N15 to N14 medium, they had a hybrid or intermediate density. So which one transferred from N15 to N14? They had a hybrid or they had an intermediate density. When they checked the density, the DNA which is extracted from the culture 1 generation after the transfer from N15 to N14, that one was having, or that one had a hybrid, or that one had a intermediate density. Okay, but the DNA which is extracted from the culture after another generation, that means after 40 minutes. First one was after 20 minutes, just soon after the transfer. Then second one after another generation, next generation formed. So that after 40 minutes, that means the second generation, that was composed of equal amounts of hybrid DNA and light DNA. In the second generation or after 40 minutes when they collected sample, so the equal amount of this hybrid DNA and light DNA. So they saw equal amount of hybrid DNA and light DNA in the second generation. Then, so that's how they proved the experiment. So that was the Michelson and Stahl experiment. That was the experimental proof. Then machinery and the enzymes. So in living cell like E. coli, the process of replication that need a set of enzymes. So the main enzymes, which are all the main enzymes? DNA dependent DNA polymerase. That is the main enzyme which is required for the process of replication. DNA dependent DNA polymerase. Note down. And that is used as a DNA template to catalyze the polymerization of deoxynucleotides. So that is working as a DNA template or template stand to catalyze the polymerization of deoxynucleotides. Then these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. This DNA-dependent DNA polymerase is a highly efficient enzyme because in a short duration, it is catalyzing, or that is catalyzing the polymerization of large number of molecules. Okay, that is very efficient and that is having high capacity, or that is having high capacity to catalyze the polymerization of large number of nucleotides in a short time. Then E. coli has only 4.6 into 106 base pairs, and that completes the process of replication within 18 minutes. E. coli completes the process of replication within 18 minutes and number of base pair 4.6 into 106 base pairs. Okay. Then that means the average rate of polymerization has to be approximately 2000 base pair per second. So how that the rate of that polymerization, that is around 2000 base per second. The average rate is around 2000 base per second. So this is not only fast, but it is giving high degree of accuracy also. This is, we can't say that it is very fast. Not only fast, not only the rate of that reaction, that is providing a high degree of accuracy also. It is accurate also. Okay. So some mistakes in this is faster. This reactions are running very faster rate, right? So during that faster reaction, some kind of mistakes also may happen. These are called as mutations. So during replication, that makes, there is a chance of mistake also. So that kind of mistakes that are called as mutations, or that changes that during replication that will result in the mistake, that result in the mutations. These mistakes may lead to mutation or changes in that genetic material, in that material constitution of that genetic material. So the replication is a very expensive process. It's not a cheap process. It is very expensive. And deoxyribonucleoside triphosphates, DNTPs, short form is DNTPs. What is DNTP? Deoxyribonucleoside triphosphate. This serves dual purpose. What are the dual purpose? They are acting as a substrate, and along with that they provide energy for the polymerization reaction. So not only providing the energy for polymerization reaction, it is acting as a substrate and it is providing energy for the polymerization of reaction. Which one is that? DNTP, deoxyribonucleoside triphosphate. It is providing energy. It is acting as a substrate for the polymerization reaction. Okay. Okay, let me complete this. So in addition to this DNA-dependent DNA polymerase, so many other enzymes are also present, or without that we can't complete the process of replication. So which are those enzymes? So for long DNA molecule, the two strands, you can see the two strands, they cannot separate completely. So for long DNA strands, it's not possible to separate completely. The entire length won't get separated. So instead of that, it will form a small area, or that in a small opening of that DNA helix. Here the whole thing is not open, only one part is open. Here up to here it is open. That part, or that DNA helix, that part is called as replication fork. So the replication occurs within a small opening of that DNA helix, that is called as DNA replication fork. That small part, small opening of the DNA helix, that is called as replication fork. It is in fork shape, that's why it's called as replication fork. So the DNA-dependent DNA polymerase, that will catalyze the polymerization only in one direction. In which direction DNA-dependent DNA polymerase will catalyze the polymerization? From 5' to 3' direction. From 5' to 3' direction only it can catalyze the reaction. Okay. So that creates some complications in the replication fork. And one strand, you can see one strand, the template strand. This is the blue color. This is the template strand. And this template strand, the template with polarity 3' to 5', from 3' to 5', and that is another one is 5' to 3'. This is 3' to 5', and another one is 5' to 3'. And the one strand, the template with polarity 3' to 5', that in that replication is continuous. In that strand, you can see replication is continuous. 3' to 5' template strand, that replication process is continuous. See, it's continuous. But in the another one, where 5' to 3' template strand, the replication process, it is discontinuous. This is not a continuous process. It is, you can see breaks here. So that is the discontinuous process. So the discontinuously synthesized fragments, here you can see, they are later joined by the enzyme DNA ligase. DNA ligase. Okay. So DNA ligase that will join these fragments later. Then DNA polymerase on their own cannot initiate the process of replication. DNA polymerase can't initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. Okay, there is a definite region in E. coli DNA where the replication originates. Okay, so at any point that can't initiate the replication process. Here, at any point, see, this is a long strand. At any point that can't start the replication process. There is particular point or a definite region in E. coli, the DNA where the replication originates. So that kind of region, they are called as origin of replication. So what is origin of replication? Where that replication starts, the point where the replication starts. That point is called as origin of replication. So it is because of the requirement of origin of replication that a piece of DNA is needed to be propagated during recombinant DNA procedures that requires a vector. Okay, during recombinant DNA technology or combining of two different DNA, recombination of DNA. So during that procedure, that requires a vector. So vector, this vector is providing the origin of replication. Mainly this vector is providing the origin of replication. Next, in eukaryotes you can see the replication of DNA that is taking place at synthetic phase of cell cycle. In which stage? Synthetic phase of cell cycle, replication of DNA is taking place. And the replication of DNA, that should be in a highly coordinated way. And sometimes in a failure of cell division after the DNA replication, that will result in the polyploidy. Or what is polyploidy? That is chromosomal anomaly. So what is that? A failure in the cell division. Sometimes the cell division that may become failure. So in that, this DNA replication also will result. The cell division, cell may not get divided. So the replicated one will be there. There will be extra DNA in that cell, but the division won't happen. So the division is a failure. In that case, DNA content will be more. So that will create chromosomal anomaly. The chromosome number of chromosomes will be more because it is getting the replication process over, but the division is pending. So in such cases, cells will be having more DNA content. So that creates chromosomal anomaly or the polyploidy. Okay. So that's all about DNA machinery and enzymes. Next class onwards, we will learn about transcription and such processes. So is it clear? Do you have any doubt? So hope it's clear. Just go through this. Thank you.